Hi everyone, my name is Lisa Hida and today I'll be presenting on application of quantifying microbial source tracking and pathogenic bacterial markers. Lake Taihu is one of the largest freshwater lakes in China and serves as a significant source for drinking water. However, due to fecal contamination, there have been investigations using existing universal associated microbial source tracking that were designed as the most suitable in identifying human and animal fecal contamination. Fecal contamination of drinking water sources, harvestable shellfish, and recreational waters is a major public health concern. It is estimated that this river collects water from 1 million habitats residing from the agricultural areas to the downstream urban cities of the northern Zhejiang province. Because the Chaoxi River is severely polluted, it is vital that these waters stay protected to decrease the exposure of humans to pathogenic microorganisms. In order to monitor pollution in environment water, FIB, or fecal indicator bacteria, has been used to access public health risks. However, FIB detection does not tell us the source or origin of fecal contamination. Quantitative PCR, qPCR-based library-independent microbial source tracking techniques have proven to be widely applicable in the study of fecal contamination in environmental samples because they can accurately quantify the host-specific MSC target sequences. They have been developed to combat the shortcoming of FIB, identifying the source and origins of pollution. Host-associated Bacterial 16S rRNA gene markers were developed for different hosts to discriminate between human and other animal fecal sources in the environment. There were 25 sampling locations that were selected covering the east and west and junctions of the Chaoxi River as seen on this slide. Samples were collected from these locations during the seasons of autumn 2014 and winter and summer of 2015. Locations selected for sampling were comprised of domestic, agricultural, and industrial areas. The water samples were collected in sterile 5-liter polypropylene containers, and sediment was collected using a sediment sampler. Samples in triplicate were transferred to sterile 50-milliliter tubes. These water and sediment samples were then transported to the laboratory on ice and were processed within 8 hours. Sediment samples were frozen at negative 20 degrees Celsius, and the water samples were filtered through 0.22 micrometer polycarbonate membrane filters and stored at negative 20 degrees Celsius prior to DNA extraction. The water samples had the DNA extracted from membrane filters and sediment samples using power soil DNA isolation kit. The count was used for initial assessment of fecal pollution at the Chaoxi River to prioritize the locations for further quantification of microbial source tracking and pathogenic bacterial gene markers. To determine the specificity and sensitivity of host-associated microbial source tracking quantitative PCR markers, individual and composite fecal and wastewater samples were used as target sources. In total, 61 fresh individual and composite fecal samples were collected from various hosts in the Zhejiang and Jiangsu provinces. DNA extraction from the fecal sewage samples were performed using the Power Fecal DNA Isolation Kit that uses IRT or inhibitor removal technology as seen on right. Individual fecal samples from animal hosts representing pigs, dogs, and cows and composite sources from ducks and geese were collected from farms, pet stores, and the backyards of households located near the Taihu Lake, Chaozhou River watershed region in the Hozhou area. There were 4 previously designed qPCR assays targeting the total human, swine, and avian associated fecal sources. The 5 quantitative PCR assays targeting STX2, EAE, LIPL32, IPAH, and MAPA genes of the specific pathogenic bacteria were selected for this study, as seen in Table 1. The 2 qPCR assays targeting the EAE gene specific for E. coli, 0157H7, STX2 gene specific for STEC E. coli, or Shiga toxin producing E. coli, The three assays targeted the LIP-L32 gene specific for pathogenic Leptospira, IPAH gene specific for Shigella spp, MAP-A gene specific for Campylobacter jejuni. All TACMan qPCR assays contain 10 microliters of TACMan Enviromaster Mix 2.0, as seen on the previous slide. 4 previously were selected for the quantification in water and sediment samples of the Chelsea River, and 3 TACMan assays were selected for detection of the whole human and swine-associated bacteroidetes, and 1 SIBR-grain GFD assay was selected for detection of avian-associated fecal markers. I will now be discussing the results. First is the quality assurance of fecal DNA and performance characteristics of MST qPCR assays. Reliable quantification and specific detection of genetic markers by quantitative PCR poses many challenges. Therefore, assays should be carefully designed to obtain maximum achievability, specificity, and sensitivity. In regards to the performance of universal and human bacteroidetes assays, comparatively, BacUni showed slightly higher total concentrations than the GenBac3 assay in all the tested samples. For human-associated assays tested were the HF1A3 SIBR marker, BAC-HUM marker, and the HF1A3 TACMan marker, and HUM2, as seen here. HF1A3 SIBR marker was the most sensitive but showed high cross-reactivity. It was found in 80% of human origin samples. It showed cross-reactivity with chicken, cow, duck, dog, and goose fecal samples, identifying it as the least specific at 69.5%. BAC-HUM marker had the highest specificity at 80.4%. The BAC-HUM assay also had a sensitivity of 73.3% and was detected in 3 different host fecal samples, chicken, cow, and dog. The HF183 TACMan marker was highly specific among human-associated assays at 80.4% and was found in 73.3% of target sources. However, the HF183 TAC marker was found in only 2 different host fecal DNA samples, chicken and dog. HUM2 was the least sensitive marker and was detected in only 53% of the samples of human origin. It was also detected in chicken, cow, pig, dog, and goose fecal DNA samples, making it less specific, 78%. In general, chicken and dog fecal samples had cross-reactivity with all of the human-associated assays. HF183 TACMan was the only assay that did not exhibit any cross-reactivity with cattle or swine samples, while the other 3 showed cross-reactivity with either cattle or swine fecal DNA samples. The animal-associated MST assays consist of the bottom half of this table 2. Pig 2 BAC was evaluated with 10 pig fecal samples, while the target was found in 90% of the samples. The pig marker was highly specific at 95% and had a low level of cross-reactivity with cow fecal samples. BAC-cow marker was found in 100% of cattle fecal samples, but showed cross-reactivity with pig and chicken fecal DNA samples. The avian associated MST, microbial source tracker, AV4143 marker, performed very distinctly on the tested fecal DNA samples. The avian marker was found in 100% of chicken fecal samples, but was also detected in human and cow fecal DNA. GFD markers were only found in 7 of the chicken feces samples and cross-reacted with dog fecal DNA samples. Data on the presence and distribution of microbial source tracking markers in water and sediment samples at 15 sample locations were determined by quantitative PCR. The total bacteroidetes concentration was high during winter for both water and sediment samples. The water samples from location 16 and sediment samples from location 15 were found to be the most fecally polluted, regardless of the fecal source. This makes sense as locations 15 and 16 are close to a water waste treatment plant. To assess the microbial quality of Chelsea River water and sediments, we take a look at HF1A3 TACMan, where the water samples collected from locations locations 12 and 16, and sediment samples collected from location 15, we see the highest concentration of a human-associated marker during the 3 seasons. Location 12 is very close to Huzhou City, while location 15 and 16 is adjacent to suburban areas and the water waste treatment plant. In water samples, human-associated markers were the most frequently detected at 97%, then avian at 89%, and swine at 84%. For sediment samples, FC, or fecal coliform counts, were higher during the summer, which made sense as warm weather increases the acclimatization of fecal coliform bacteria. Human-associated MST markers were detected more than often at 86% than swine at 64%. Avian markers were positive for several samples, but always below the limit of detection. Fecal contamination via the swine markers were observed in water samples at location 1 during summer and in sediment samples at location 10 during winter. Location 10 was closer to a rural area where the agriculture inputs to the river stream. Avian-associated fecal pollution was found to be the most second-dominant host-associated fecal pollution in the river water samples at 89%. For sediment samples, avian markers were detected in only 4 of the 15 samples collected in the summer season. Highest avian marker concentration was observed at location 6 for both water and the sediment samples, which makes sense as location 6 is a suburban area near Quita village where there are many swine and poultry farms located. Data on the presence and distribution of gene markers for 5 bacterial pathogens can be seen on Table 6 as well. Data on the presence and distribution of MST markers in water sediment samples at 15 sampling locations were determined again by quantitative PCR. Respectively, Campylobacter jejuni and Shigella spp, Campylobacter jejuni at 62%, 53%, Shigella spp at 60% and 91%, followed by STEC 55 and 51%, and pathogenic Leptospira 33 and 13%. E. coli marker was detected only in sediment samples of 11%. MAP-A gene was present in quantifiable range in 20 out of 45 water samples and 13 out of 45 sediment samples. The highest MAP-A gene concentrations observed in water samples collected at location 16 and in sediment samples collected at location 20. However, in the case of Shigella spp, although the IPAH gene was detected in several samples, it was quantifiable in only 2 out of 45 water samples. IPAH gene in the case of Shigella spp was only quantifiable in only 2 out of 45 water samples. Sorry about that. In sediments, IPAH gene was quantified in 14 samples with highest concentrations observed at the location 12. STX2 was quantified in only 2 out of 45 water samples, locations 5 and 21. In sediments, it was quantified in 14 out of 15 samples with the highest concentration observed at location 14. LAP-L32 gene, pathogenic Leptospira, was quantified in 15 out of 45 water samples, was quantified in 6 out of 45 sediment samples with the highest concentration observed at location 20. EAE gene was quantified in only 3 sediment samples with the highest concentration recorded at location 20. Now, the correlation between these FC MST markers and genes of bacterial pathogens, where the concentrations of FCFIB were positively correlated with BAC-UNI and HF183 TACMAN, but not with pig-to-bac and GFD markers. This indicates that the common source of fecal pollution is from water waste treatment plant or human waste entry into the river. BAC-UNI markers showed a strong positive correlation with HF183 TACMAN. This suggests humans as a major contributor to the total Bacteroidelis content. Bacuni also showed positive correlation with STX2 gene. HF183 showed negative correlation with GFD marker and positive correlation with STX2 gene. Multiple fecal sources, such as pig, cow, and poultry, of STX2 gene presence in the environment. No significant correlation was observed for pig tobacco and GFD markers with the marker genes of bacterial pathogens addressed in this study. To conclude, MST, microbial source tracking, qPCR, quantitative PCR, was the most suitable for differentially identifying and monitoring human and animal fecal contamination in the Tahoe watershed. These human, swine, ruminant meaning cow, deer, etc., and avian markers, along with marker genes specific to selected bacterial pathogens, were quantified in water and sediment samples via the Chowchi River and collected from 15 locations. Based on fecal coliform enumeration and samples collected from the 15 locations that were used for the quantification of MST and pathogenic bacterial gene markers, we confirmed the presence of fecal contamination. Human-associated markers were frequently detected at several locations, 3, 12, and 16, with 16 having the highest concentration on one or more occasions, indicating that they are major human fecal contaminated sites of Tiaoxi region. Swine-associated markers were frequently detected in samples from locations 13 and 21, and the avian-associated marker was detected with a high concentration at locations 6, 15, 20, and 21, matching the land use pattern and pointing to the entry of swine and avian fecal sources to the Tiaoxi River. Among 5 bacterial pathogens monitored for avian, Campylobacter jejuni was frequently detected at locations that are primarily polluted with avian fecal material, locations 4, 6, 8, and 13, and high concentrations were detected at locations 6, and 13, posing human health risk. Shigella spp was frequently detected at higher concentrations in 2 locations, 3 and 12, that are highly contaminated with human fecal sources. And lastly, Shiga toxin-producing E. coli was found at 2 locations, 5 and 21, which are contaminated with either human or pig fecal sources. And that is all for my presentation. Thank you for listening. Please post your questions, and I will get to them as soon as possible. Thank you.